Welcome to April's BMS Talk, and it's my great pleasure to introduce our chair for this evening. Mike Chukai has spent most of his career in industry working on the discovery of novel disease control agents, including more than 20 years at Syngenta, and now he works at Biotalis in Belgium. So thank you very much, Mike. I'll hand over to you.
Okay, thank you very much for the introduction, and I'd like to welcome everybody and thank you for taking the time to join us on our webinar this evening. And we've got three hopefully terrific speakers to give us a talk each tonight, all of whom are at the other end of the career than mine, so very new and some exciting science tonight, I hope. So we're going to have three talks. One from Alex Pate, one from Lisa Humbert, and one for Jonathan Heel in that order. And I will introduce them as we go along. First of all, if you want to ask questions, please do. Please use the chat, not the Q&A button. If you're not familiar with Zoom, just move your mouse and it should appear at the bottom of the screen and you'll be able to open it and type any questions, and I will field those questions to the speakers as we go along. I think we will just move on to our first talk then. So, Alex. So Alex is a third-year PhD student working in the School of Life Sciences at the University of Nottingham. He studied for his BSc in Biological Sciences at the University of Surrey. And during that time, he had an opportunity to work in the microbiology team at GSK Consumer Health, now Helion. And Alex's academic and industrial experience has fostered some real interest in microbiology, fermentation, strain improvement, and really how you exploit microbes to make beneficial products. And he's going to talk to us about the fungus that is used for growing corn. And so the title of his talk tonight is Meddling with Microprotein, Novel Strain Development of Fusarium venenatum. Over to you, Alex.
All right, thank you. I'll just share this now. Hopefully, can I just get a double check that the title slide is visible?
Yes, we can see that. Thanks.
Brilliant. So thank you everyone for tuning in. My name's Alex, and I'm going to be taking a bit of time to talk to you about my PhD project that I'm studying here at the School of Life Sciences in the University of Nottingham. And so, meddling with mycoprotein, novel strain development of the fungus Fusarium venenatum. What's that about then? So I think I can start by going about half a century back in time for a bit of background. And so consider that back in the 60s and 70s, many experts were panicking. They were predicting that widespread famine was imminent because within a few decades, our population would just be too big, too many mouths to feed. And the Rank Hovis McDougall conglomerate of cereal companies, headed up by Lord Rank, they were interested in finding a microorganism that could convert simple sugars and incorporate it into its biomass, and then that biomass would be safe to eat and high in protein, and so nutritious. And after searching, taking samples from locations all around the world, and screening thousands and thousands of organisms, an ideal candidate in the form of Fusarium venenatum, a filamentous fungus, was found. Now, I guess the catastrophe of famine worldwide didn't quite come to fruition. However, it's still worth considering that even in the 2020s now, hundreds of millions of people around the world suffer from undernourishment. And I guess quickly to go over how mycoprotein is made, if Fusarium venenatum is used to make it. And essentially, the production process, it's been upscaled quite a bit since the fungus was first identified as a good candidate. In a giant airlift fermenter full of growth medium, it's inoculated with a bit of the fungus, and then a mycelium of fungus develops, and it's circulated around this fermenter tank, powered by the hydrostatic pressure of oxygen, ammonia, and glucose all being pumped in. The medium is continuously recycled so that there's always enough glucose to sustain the growth of the fungus continuously. And as the mycelium develops, this mycelium is made up of lots of tiny filaments. These are called hyphae. This mycelium is tapped off and processed in such a way that all those hyphal filaments then squash together, and you get a product at the end of it that has a texture somewhat similar to animal meat. And that is what mycoprotein is. And it's used by Quorn in their line of meat alternative products. So this process needs to be terminated every 6 weeks, because if the fermenter is run for a longer, you sort of get little instances of evolution happening in the fermenter over time, purely by chance, where randomly little branches of the mycelium in the fermenter will there'll be mutations randomly happening, and occasionally it will mean that particular branch of hyphae branches more often as it grows and extends, and so it can utilize the growth medium in the fermenter better than the mycelium all around it, and so it quite quickly outcompetes the rest of it. And as that accumulates and happens over time, the more branches that form in that mycelium disrupt that all-important meat-like texture that makes mycoprotein usable for meat alternatives, and so the fermenter must be stopped, cleared, and restarted. And there's you lose several days in downtime for that. Also, the current sugar used is glucose, as I said. Glucose is quite expensive, and it has to be delivered daily as a highly refined syrup. To save money and make it easier to store, switching to a sugar like sucrose would be ideal, and mitigate a lot of these challenges. However, there are then challenges associated with doing that, including the fact that you can actually get this fungus to start producing toxic byproducts if you mess around with the process too much. Oh, and for reference, these airlift fermenters, when I say they're big, they are about 20 stories tall. They're very big. And yeah, so to summarize, the current production strain, and that's designated A35. That's what I mean when I say A35 going forward. A35 produces a safe, nutritious, meat-like mycoprotein with the current production process. The fermenter uptime is limited, and adjusting the process risks compromising the safety and or the texture of the product. And with competition in the alternative protein space booming now more than ever, and the ongoing importance of climate change mitigation, there's great incentives to start finding ways to future-proof this production process. Because also one thing I think I actually forgot to mention was that mycoprotein has quite a few benefits over traditional meat. Certainly stuff like beef, in terms of less land use, less water use, and less CO2, while also being a relatively high protein food, low in carbs and fat, and with more fiber. So pretty good. And so that's where my objectives for this project come in. So broadly, I can sort them into two objectives. The first being characterization of pre-existing different strains of Fusarium venenatum to try to identify if a candidate superior replacement strain already exists. And then the second involves attempting to utilize some biological methods to produce new strains of Fusarium venenatum that might make better candidate production strains. So we'll start with objective 1 first. We've got 4 candidates selected due to their characteristics, due to their relevance in the production process. So that's conjugation rate, mycotoxicity, protein content, and hyphal branching. I'll expand on all of those as we go. So to start, in general, for pre-existing strains, we have a couple of dozen strains of Fusarium venenatum here at the University of Nottingham. They've been procured from various culture collections, as well as very generously as gifts from various colleagues in the industry. Interestingly, some previous attempts to identify new strains of Fusarium venenatum in the environment weren't very fruitful. And so it would appear this is actually quite a rare species and potentially not as ubiquitous as people originally thought. But in any case, for the strains we have, we whole genome sequenced all of them, and preliminary analysis did identify some species-wide genetic variation. In terms of other early work, some of the work I did previously showed that there were disparities in the ability of each of these strains to produce conidia. And I'll talk about conidia because they're quite important. They are essentially just fungal clonal spores. So they'll bud off as the mycelium grows. And they have a much slower metabolism than the rest of the mycelium or from a fungal hyphal filament. They're produced in, for instance, times of environmental stress, and they remain dormant, and they'll either hunker down or get dispersed to a new area. And then when conditions become favorable again, they can germinate and start growing into a new fungal mycelium. But it's those hyphal filaments I was talking about earlier that make that meat-like texture that's really important for the final product. And so more conidia, less hyphae, bad for the product. So that's something we want to look at. Also, some early work by previous PhD student Jack Whitaker showed that some of the strains of Fusarium venenatum produce DAS, and this is a trichothecene mycotoxin produced by many species of the genus Fusarium, which is a genus that actually has quite a few prolific plant pathogens in it, mainly because of their ability to produce these trichothecene mycotoxins. We'll get back to that in a minute, because I first want to talk about conidiation again. So I said I want to compare the conidiation rate between all of these strains, and I can do that quite easily by growing up each of these strains in a small flask, filter out the large bits, and then count how many conidia I can literally see under the microscope, work out concentration from that. I compare all of those numbers, those calculated conidiation rates, and I can color code them based on whether they conidiate significantly more than A35, red because, again, more conidia, bad in this case for the product. Green if they conidiate significantly less, because that's good for the product, less conidia, more mycelium. Or if they're not significantly different to A35, the current production strain, that's yellow. It's so-so, basically. And we can see here that most strains are similar to A35. One's quite a bit higher, but we do actually see a few that are conidiating significantly less, which is quite promising. So back onto the mycotoxicity. So DAS production profiles differ between the strains. We've already established that, and with sugar feedstock using either glucose or sucrose to feed the fungus. But I also said that DAS is just one mycotoxin of a roster. And we want a method that can quickly test all of these strains for mycotoxic activity without just narrowing our search down to one at a time. And so we've picked a yeast, Clivoromyces marxianus, that's shown to be sensitive to a broad range of mycotoxins. And I can grow our different strains of Fusarium venenatum up in flasks, harvest the liquid medium they've been grown in and exposed to and leaching compounds into, or literally extract and redissolve organic compounds from the mycelium itself. And then I can grow this sensitive yeast in the presence of these solutions. And then I can measure how fast that yeast grows when exposed to the solutions and use that as a proxy for how toxic those solutions were. And so the toxins the fungus was producing under those conditions. So here's a good place to start with just A35. So this is a graph showing the growth of the yeast when treated with various solutions. We can start with the untreated yeast over on the left-hand side. That's that black shaded line. And that curve shooting up quite early on. And it shows that the yeast is growing quite quickly. It's nice and happy. Worth mentioning, these curves are a combination of 9 curves averaged out, and then shading either side of that average line is from the variation of each line around that average and how accurate that average is. So back to the actual curves themselves. The red curve, that's the yeast growing when exposed to A35 when grown on glucose supplemented medium. So that's the sugar used in the current production process. You can also see that curve is shooting up quite close to the untreated control. It looks like it's slightly pushed back on the graph, but then when I compare the actual growth rates at that time when the control is growing at its fastest, they're statistically indistinguishable, and that's what we'd expect in this case. But then if we compare A35 grown on sucrose supplemented medium, that yeast is growing tens of hours later. And so there's clearly some sort of toxic effect happening here. This is another interesting strain of Fusarium venenatum, actually. So this is a different strain to the production strain. The color scheme is broadly the same for the two differences. If you're looking for those red and blue lines for glucose and sucrose grown media, respectively, well, they're both down at the bottom. The yeast isn't growing at all, in this case, at any detectable level within the confines, the time constraints of this assay, regardless of the sugar used to grow it. And so there was toxicity wherever. This is a more toxic strain, it seems, than the current production strain. But then we have a strain like number 3 here, and you'll notice that those red and blue curves are both way over on the left-hand side with that untreated control curve. And the growth rates here again, when measured, are statistically indistinguishable from the control, regardless of sugar used. And so this is quite interesting as a potential strain that doesn't have that increased toxicity on sucrose that some of the other strains have. I could show more graphs, but I think I'll stick to a summary table again for this, a color-coded summary table. Red strains here, they produced measurable toxic effects regardless of the sugar feedstock used. Green strains that you can see, a few of them, they did not produce any measurable toxic effects within the sensitivity of the aforementioned test I was using. And yellow strains, including A35 in this case, show toxicity on sucrose only, but none on glucose. And this was just testing the liquid medium that the fungus was originally growing in. The other option I've got to stack up later on, and I'm in the process of doing the final data collection for this, is testing the extracted and concentrated organic compounds in the mycelium itself. And when it's done, it'll be a nice supplement, certainly for those so far promising green-looking strains. Sadly, no green across the board strains so far. We have some greens, some that are green and yellow only, which is it's certainly promising. And as I say, depending on what we were expecting, it might be as good as we'll get. But it is also worth noting that there will be more characteristics in this table coming up. And so those two I mentioned, as I briefly mentioned them earlier, that was the protein content and the hyphal branching. And as I say, if the protein content varies between the strains, that's something that would be very important for the nutrition of the final product. And also, if they branch differently when they're growing, I've already spoken about how important that low rate of branching and the texture in the final product is down to the hyphal filaments growing as they are. And so more or less branching will have a significant impact on the final product. And also, it's worth mentioning that with those whole genome sequences, I do want to go into a bit more detail in those as well, see how strongly some of these characteristics, the differences there, are linked to the genome. Okay, so I'll finish up objective 1 there. Can move on to objective 2 now, which is making novel strains, and it's where I get to talk about sex. So fungi can have sex too. So far, when I've been talking about Fusarium venenatum growing by, it's just been copying itself over and over as it grows. But some fungi, not all, have different mating types and can actually sexually reproduce with compatible strains of the same species and produce offspring with different characteristics, combinations of characteristics from both parents. Some fungi can have sex with themselves. We're not going to talk about that for the purposes of my work. But in general, a sexual life cycle in Fusarium venenatum specifically hasn't been seen so far. It's been seen in some other closely related species of Fusarium. And so theoretically, if I can find the right pairing of strains and the right conditions, I might be able to spot those hallmarks of sexual reproduction, which would be fruiting bodies. In the case of Fusarium, these fruiting bodies are called perithecia. And so far we have 2 mating types identified in Fusarium venenatum. So we can call that heterothallic. It can't have sex with itself. It can only outbreed with compatible opposite mating types, strains of the same species. And in general, this would be fantastic because it would mean you can make new strains of the fungus without any genetic engineering, which is a big no-no for food products in general in many places. And it would also give us the opportunity to crossbreed strains that maybe have some favorable characteristics, maybe some not, and hopefully select for some offspring that have the best balance. And yes, so before any sexual crossing can be attempted, we need to find the mating types of each strain in our collection. And then we have a few different methods of introducing mating type compatible strains together. One involves mixing conidia of both mating types together on a petri dish, and then waiting for it to grow. Another involves inoculating different sides of a petri dish with different strains, waiting for them to grow, and then checking where the different mycelia have come together, see if we can see fruiting bodies there. And then another method involves growing one strain up and then rubbing it with a suspension of conidia from the opposite mating type onto it and leaving it for a while to see if they do their thing, I guess. And you can see we have mostly mating type 1 strains in our collection, but we do have 5 mating type 2 strains, and that is frankly plenty possible combinations, if I do say so myself, with repeats of each mating combo and each combination on a different media type. That's thousands of individual petri dishes to check. As a reference, this is an example of what Fusarium perithecia look like under a microscope, so I'm always on the lookout for something like this, essentially. I haven't found anything like that yet, unfortunately, and I still have lots of plates to check through. I'm still holding out some hope, but this is not the only method of generating new strains. There's some evidence that diploid strains of Fusarium innatum mitigate some of the aforementioned issues of mutant accumulation in the fermenter over time. Current strain is haploid. A diploid strain would mean the fungus has 2 copies of each chromosome. And we're looking at ways to generate diploid strains in Fusarium innatum. But also, as a tangentially related life cycle, I guess, sometimes when 2 strains grow together, even if they don't undergo sexual reproduction, they can occasionally spontaneously merge and become a heterokaryon. So that would be a mycelium with the nuclei of 2 different strains in it. And then very rarely again, if that happens, very rarely those nuclei in close proximity will merge and form diploid nuclei. They're not usually very stable, and they will often go through a series of erroneous mitotic cell cycles before then being stable haploid nuclei and strains again. But those errors in the mitotic events mean that the haploid nuclei at the end of the process are different than the ones that went in. So no sex, but different nuclei. Different nuclei mean different strains, potentially with different characteristics. That's parasexual reproduction. Okay, so I think this second objective was a bit heavier and a bit more theoretical than objective 1, partly because I have fewer obvious results to share. The negative results for sex are a shame, but we still have lots of opportunities to see that. With constraints, I'm going to try and stack up and combine some of the reps I'm doing for that in the future to try and get more done at once, using up less space in my incubators. I'm also looking at crossing attempts and for the diploid and parasexual work, because both hinge on me being able to identify diploid, hyphae, and conidia when they arise. A way of actually identifying these is being explored. Unfortunately, Fusarium particularly are annoying for that, because their conidia are irregular shaped and they have more than 1 nuclei in them anyway, usually. So we have some options we're exploring. I'm excited to share those results as I go forward. But that'll be all from me for now. I hope I didn't go too much over time there. I want to thank my project sponsors, the BBSRC, and of course Corn Foods, my supervisory team, our group's postdocs, and everyone that helps make the life sciences fungal biology group here at the University of Nottingham vibrant and interesting place to work. And oh yeah, and I'd like to thank all of you as well for watching.
Well, thank you very much. So I'm sure people are virtually clapping as well. So thank you very much, Alex. I'd just like to remind people, if you have a question for Alex, please type it into the chat, and we'll have some time to ask. But given that there's not a large number at the moment, I will kick us off. So, I mean, I can understand why you're trying to get the sexual crossing to work to generate a wider panel to look for attractive traits. But have you specifically thought about actually mutating and looking for lower conidiation rates, for example, using the existing strain? So strain 3, for example, it ticks 2 of the boxes, but it seems to have a high conidiation rate. Could you actually mutate that and then do selection for conidiation to get a more attractive strain from that, or is mutation a bit of a no-no in Venenatum for food?
Mutation would be okay. I think for this, it was, I guess you could say, the interest in learning about the potential sexual biology of Fusarium venenatum in itself is an interesting academic and research prospect. Anyway, I'm not sure if there's other work going on mutating strains of Fusarium venenatum. I wouldn't be able to comment on that. But as I say, with the existing strains we already had, since we had a couple of dozen, it made sense to prioritize first just seeing if there was already a good strain in that respect, and then looking at the those other natural methods, I guess you could say, for introducing changes ourselves.
Yeah. I mean, obviously, if you can get the sexual crossing, that gives you a much wider range of things to look at, and you can do all sorts of selection. But if it becomes very difficult to find it or stimulate the sexual crossing, then I guess that's a backup, I suppose. So we have a question that's just popped up. Are you screening for mycotoxins other than trichocytines? I can't pronounce that. Fumonazins and ergot alkaloids.
It's a good question. At the moment I can't remember specifically from the reading I originally did when sort of setting up this study exactly which other mycotoxins the yeast we're using is sensitive to. At the very least, it's sensitive to trichothazines, and that was obviously early on identified as an important group to target. And since this yeast assay gave us a way to quite quickly and broadly target a large group of related trichothazines produced by Fusarium, that was a nice springboard starting point. Obviously, for the strains that do come up more promising, we are looking into doing more specific screening for certain types of toxins, and actually looking to see specifically what is being produced and see if they are, say, green in our metric, whether they are actually green for everything that would need to be considered for an actual food product.
Okay, let me see if there's any more questions. One question would be, so in terms of you've looked at the impact of using glucose and sucrose on the on the production of mycotoxins. Does that have a significant impact on the propensity to actually generate hyperbranching and or conidiation at other stages?
Sorry, I've opened the chat and I just watched a message coming. Would you be able to repeat your question, please?
What okay.
No. I read it and so I missed the first part of your question. Could you?
Right. Okay. Yeah, I was just asking if you've done the screening with sugar. So instead of glucose, so you did your testing of glucose versus sucrose and the impact of that on mycotoxin production. But does it have a significant impact on the different strain productions or propensities to generate the hyperbranching mutation and or the conidiation effect as well?
Specifically for conidiation, I can't answer that question. I do believe some preliminary work done that originally had sucrose identified as a potential not only cheaper but also better sugar source. If I remember correctly, was that it might have been less mutation in the fermenter. I'm cautiously saying this because I can't specifically remember whether that is true or if I'm conflating memories. It's been quite a while since I looked at that specifically, but yes,
Okay.
there are multiple characteristics can change with the different sugar feedstock.
Okay. Got a couple of other questions on the chat. So M asks, are microproteins considered to be ultra-processed foods?
Yes, they are.
Okay. And then Chloe asks, what method did you use for GWAS? Do you plan to look at possible chromosomal rearrangement SNPs alongside the strains, which could explain the different phenotypes observed?
So we've not done the GWAS yet so far. We have some preliminary mapping rates based on Illumina platform whole genome sequencing. And that gave us some rougher percentages, but more in-depth bioinformatics hasn't been done yet. We only have those some very rudimentary mapping rate differences between each strain and a reference strain. In terms of the things that we then want to look at, I guess I spoke a bit about the characteristics. Mainly, another thing that I didn't actually get to talk about that would probably be more of a focus for the bioinformatics and looking at differences in the genome would be sex-related sex factors, essentially genes in the genome responsible for producing pheromone precursor molecules and then pheromone receptors, so that we could then have an idea of like what's going on there, and that could also be linked to expression work as well as what genes are actually there. But yeah, obviously, depending on the characteristic we're more interested in, for toxicity, we can look at genes known for producing different, say, trichothecenes, and look at that as well. I say I'll be able to answer more questions on that when we actually get started, and that becomes my focus for a couple of months.
Okay. And one last question, because we're going to have to move on. Maria points out that in her experience with a range of Ascomycete sexes, a response to stress. So have you tried stressing the mycelium, e.g. paresthesia often arise as an edge effect, for example?
So far, we've been testing media types that have worked for other Fusarium in the past. So things like Cara agar and VA agar. And we've been stressing them insofar as we've been incubating them under cool white fluorescent light. We've not been doing that under UV, and we haven't been using minimal media either. That is something we're potentially looking at going forward as another media type, would be to grow the fungus up on a nutritious media at first, and then maybe transfer it using a nylon sheet onto a minimal medium, stress it out, then see if we get those fruiting bodies. So yeah, a good point.
Can often be a starvation response. Yeah. Okay. Thank you very much, Alex. Thank you for a very fascinating talk and fielding the questions. I'm afraid we have to move on. So next speaker is Lisa Humbert. So Lisa is also working at the University of Nottingham on a project with Rothamsted, but prior to her PhD studies, she studied for an MSc in chemistry of biomolecules at the University of Montpellier. She later worked in R&D as a synthetic chemist in a pharmaceutical company in Brussels, in Belgium, before relocating in 2021 to the UK and starting work on a much more biological project, which is being funded by Future Foods, Beacon, and is the basis of her PhD program. So really trying to understand whether some of the sex hormones could be used for plant disease control in a joint project between Nottingham and Rothamsted. And the title of her talk is Sex as a Weapon, Using Fungal Sexual Hormones to Control Plant Diseases. So over to you, Lisa.
Well, thanks, Mike, for the introduction. Can you see my slides?
Yes, I can. Yes.
Perfect. So first, I would like to thank the BMS for giving me the opportunity to present my research area today. So as Mike already introduced, I'm a third-year PhD student from the University of Nottingham, but my PhD is mainly based at Rothamsted Research, which is an agricultural research centre located near London. So in my PhD work, I study the sexual hormones of fungi and how we could potentially use them to control the spread of plant disease. So I'll start by giving an introduction so you can know what's behind this mysterious title, and I will talk a bit more about my research after. So I think we'll all agree here to say that fungi are incredible. They're amazing in terms of food. They taste amazing. They make great cheese, and they are also they contribute to healthy soil and healthy plants. But I apologize, today I'm going to be talking about the bad fungi, the plant pathogens. So I've put some examples of fungal disease here. I think probably the most famous one would be the one caused by the Fusarium species on banana plants or wheats, but there is also different species of mildew, rust disease, septoria disease, lots of them. So they are economically important pathogen because they cause yield loss in our crop, but they're also very difficult to control. So the current disease management for those fungal diseases are usually the use of broad spectrum fungicides. The issue is that you might be aware that fungal diseases are becoming more and more resistant to broad spectrum fungicides because we overuse them. But also they are not good for the environment, they're not good for the soil, the insects, and for human health. Another disease management technique is the use of resistant cultivar. But here we have a similar issue that the pathogen are adapting to the resistant cultivar, and it also reduce the diversity of the species of plants that we use for our food. So it's becoming more and more urgent to find new way to tackle fungal disease. So the crop that I'm working on is oilseed rape. So it's a crop that is widely produced across the world because we use it mainly to make cooking oil for human consumption, but also animal feed and biodiesel. So as you will see from the map, the main producers of oilseed rape are China and Europe. And in the United Kingdom, we produce around 1.5 million tons of oilseed rape every year. Unfortunately, lots of the yield is lost due to fungal disease, and it's estimated that around 25 to 30% of yield loss annually is caused by the disease called light leaf spots. So you will see, once again, from the map, you see the disease is widely distributed across the world, and we find it on 4 of the 5 continents. So the light leaf spot disease is caused by the fungal pathogen called Pyrenophysiza brassicae. And so the symptoms on oilseed rape and on brassica crops in general will be some lesions that starts appearing on the leaves and the stems. And it's going, once the fungus is established, it's going to disturb the natural growth hormone of the plants, and we'll see some distortions of the leaves and the pods being used to make the oil. So that's what's causing the yield loss. So it's the main fungal pathogen of oilseed rape in the UK, but we also find it on different brassica crops, so Brussels sprout, cabbage, cauliflowers, etc. So in order to tackle the disease, we need to understand the life cycle of P. brassicae. So during the crop growing season, which is most of the year, the pathogen is going to reproduce asexually. So it's one individual that is going to make asexual spores, and those are called conidia, and they are made using those asexual structures called the conidiophore. So the conidia would be spread by the rain, and they're going to contribute to spread the epidemics on the plants. So for the rest of the year, so the off-season, the pathogen is going to reproduce sexually on the crop debris, and is going to reproduce sexually in order to survive during the summer. So the sexual reproduction, we will have two mating types that complement each other in close proximity with each other, and they will produce those sexual structures. So that's the round blobs that you see. You can see it better in here on the microscopy. So those sexual structures are bearing the sexual spores. And yeah, they're going to release the sexual spores. And so this sexual cycle is going to be ruled by the sexual hormones. So there's different types of sexual hormones that are being produced during the sexual cycles, and they all rule different mechanisms within the fungi. So my work is building on the previous work from Alice and Ashby. So we decided to look at the sexual hormones that are called the sex factors. And why are we interested in them is because the sex factors are believed to be responsible for inducing the switch from asexual to sexual reproduction, but they also trigger the synthesis, the formation of those sexual structures, the apothecia. So we hypothesized that those sex factors would suppress the asexual sporulation and potentially suppress fungal growth. And the idea is to try and use those sex factors as novel antifungal compounds for the light leaf spot disease. So to test our hypothesis, we extracted the sex factors with organic solvents. We end up with a liquid extract containing the sex factors, and we apply them on cultures of P. brassicae. And what we see after 15 days is that the plates start darkening. And you will see that the more we increase the concentration of the sex factors that we apply on the plates, and the more darkening we have. And when we count the number of asexual spores, we see that this darkening is directly correlating with a suppression and inhibition of asexual sporulation. So on this graph, it's a logarithmic scale, so we can't really see it, but the reduction in asexual sporulation is quite significant. We're going from, on the control plate that didn't receive any treatment, we have around 110 to 6 spores per milliliters, and we're going down to 10 to 6 spores per milliliter, so it's quite significant decrease by 97%. So that's where my PhD project comes in. So the idea is to try to investigate the use of the sex factors as a novel management tool for light leaf spot disease. And I actually worked in lots of different disciplines of science to try to tackle the question through a different point of view. So I've been using a lot of microscopy imaging because we're trying to understand the morphogenesis of the fungi when it's exposed to the sex factors compounds. I used analytical and synthetic chemistry because we want to purify and characterize the sex factors compounds so we can use them in the field. I used a combination of bioinformatics and molecular biology to try and understand this biosynthetic pathway of the compounds. And finally, as a proof of principle, we did some implanted trials. So today I'm only going to talk about a couple, like two of the parts of my PhD project. So to start with the microscopy imaging. So as I just said, we had up until here, we had only been looking at the culture when they have been treated with sex factors after 15 days. And what we see is very low numbers of asexual spores and a darkening of the plates. So I realized an experiment where I was monitoring the asexual sporulation during 15 days every day. And so there's two key points that we see during these experiments. So the treated plates are in orange and the control are in yellow. So you will see on the treated plates that it has an interesting pattern. First, we thought that because we would see very low numbers of asexual spores after 15 days, the culture would only produce very low numbers of spores, which was not the case. So they were actually sporulating, even with the sex factors treatment. And there is a very interesting change in like a switch in the sporulation pattern around day 12 and day 13. And the second things that we notice, so as you've seen before, the plates after 15 days are completely dark. But we noticed with using light microscopy that they were actually starting to turn black after 11 days post-treatment with the sex factors. So from that experiment, we started getting more interested in what happens between day 11 and day 13, what happens for it to switch the sporulation pattern. So to investigate that, I reproduced the experiment, but this time I looked at my plate using scanning electron microscopy, which is, for those who are not familiar with this, it's an instrument that produces an electron beam and is going to give you information on the surface topography of your sample. So to go through quickly with the controls, so that's how my fungus is supposed to grow normally. So it's just producing hyphae, and then we see some very nice conidiophore producing the asexual spores. And then from day 11, we see just the culture completely covered in spores, blankets of spores, that keeps going through until the end of the experiment. So at the end of the experiment, you still see some very nice conidiophore structure producing spores. But what's most interesting is what happened with the culture that have been treated with the sex factors. So as we expected, for the first 10 days of the experiment, no change. We still see some very nice conidiophore and they still produce spores. And that's when it gets interesting at day 11. If you remember, that's when my culture started darkening. We could actually see a biofilm that starts appearing. So it was very sporadical at this time. It starts appearing here and there on different points of the culture and start growing some mycelium on the biofilm. At day 13, this biofilm had expanded and they were covering completely the culture, and a dense mycelium had grown back on the biofilm. And by day 15, we could barely see any asexual, any conidiophore, so any asexual structures, but the one that we were seeing appeared to be deformed, like have deformation. They looked quite hollow and wrinkled. I don't really know how to describe them, but they look like mutant spores. So I'm in a process of looking at the viability of those and see if they are able to germinate or not. But what we learned from this experiment, so it's really interesting, is that when we apply the sex factors, those hormones on the fungi, so it starts giving the sexual response, and this response is to produce a biofilm that is most likely made of melanin. This is giving the dark color to this film. And this is most likely a defense mechanism of the fungi that they start producing when they are sexually reproducing. So this biofilm is going to expand and they trap the spores underneath, and that's preventing the spores from being released. And that's why we barely see any spores by day 15 of the application. So that's interesting because we modulate the natural life cycle of the pathogen, and we use its own defense mechanism to against itself. So to move on quickly with the chemistry, I don't want to bore you too much with chemistry. So as I said, we extracted the sex factors, we extract them with organic solvents to get the more lipophilic compounds. And then we use a technique called liquid chromatography to purify the compounds, because of course when we extract them we end up with lots of secondary metabolites. So liquid chromatography, for those who don't know the technique, is a column. So it's a stationary phase, and when we load the sample, it will have, depending on the polarity of the compound, they will have different affinity with the column. So the compounds are the more polar are going to be eluted first, and the more apolar compounds are going to be retained on the column and be eluted last. So from that, we divide the elution into different fractions, and then I test the fraction on bioassays, and the one that are responding, so the one that are darkening, the one that contain the sex factors. So we take back this fraction and we load it again on the HPLC. We change the method to optimize it, and we fractionate it again over and over again until we're happy with the number of compounds that we have inside. And just to show you, so this is the crude sex factors extract just after I extracted it, and all of the peak that you see, each one are a compound, so there are lots of compounds inside. And after only 3 rounds of using the fractionation on the HPLC, we can see that we have a significantly less compounds inside the active fraction. So once we are happy with that, we use LCMS, so it's the same technique. There is liquid chromatography, but we couple it with mass spectrometry that will give us information on the mass of our compounds and the elemental composition. So what you see here is a plot of the compounds that were into my sample. But what's interesting is that we notice that those mass and the property of those compounds are similar to a certain class of oxygenated fatty acids. So from that, we were wondering if the sex factors, the precursor of the sex factors would be fatty acids. And if you remember, I mentioned at the beginning of the talk that one of the functions of the sex factors are they are promoting the growth of apothecia, the sexual structures, which looks like this, the wrong thing. So I realized an experiment when I was feeding my crossing cultures with different fatty acids of different length and different in saturation at different concentration. And I was looking, counting the number of sexual structures being produced, because we would think that if those are precursor from the sex factors, then the more we feed the culture with fatty acid, the more they will be producing apothecia. So here are some of the results. It's a lot of data, but what you will notice or should notice is that so when I looked at the number of mature apothecia being produced, those were increasing with when we were adding linoleic acids on the culture, and the number of immature apothecia were increasing when we were adding linolenic acids on the culture. And what's the common point between those two is they actually both have a C18 carbon chain on the fatty acid. The only difference is that they have linolenic acid that has one more in saturation. So from this experiment, we conclude that the C18 fatty acids are promoting the development of the sexual structures in P. brassicae, and they could potentially be a precursor for the sex factors. So that's a bit of chemistry. I'm going to go quickly over that because I'm quite aware of the time. So one possible thing that to confirm that hypothesis would be that we synthesize labeled fatty acids, and then we feed the crossing plates with those, and then using the same technique with the mass spectrometry would be able to see a shift of 2 units in our data if the sex factors are being produced from the fatty acid that we fed them with. So I'm not going to talk about the other work that I'm doing during my PhD, but we've done in-planta experiments in the glass house to prove as a proof of principle. And I'm also working at the moment in molecular biology. So we've identified 2 genes that have been identified in other fungi for producing sexual hormones of the same kind of structures. So we're in a process of knocking those out to try and confirm that they are linked with the sex factors. So yeah, to summarize, those sex factors produced by P. brassicae, they are able to repress sporulation. And they could be a very interesting new strategy for the control of likely spot disease because, they reduce the spread of the epidemic through asexual sporulation, but indirectly sexual reproduction as well. So compared to fungicide, they are better for the environment because they target one specific disease. They are active at low concentration, they degrade naturally in the environment, and also, yeah, they would tackle the resistance issue that we have at the moment. So on the wider aspect, it's really interesting. It would be really interesting to study if those sex factors or sexual hormones could be used in different fungal pathogen, whether if the structures is conserved in different pathogen, or if they produce similar hormones that has similar effects to the sex factors. So yeah, to finish, I would like to thank my supervisor, David, Paul, Mike, and Matthias for their help during my PhD. I would like to give a special thanks to Felicia who has been teaching me everything I know about molecular biology, but also Suzanne, who has been helping with the design and the statistical analysis of my glasshouse experiments. And thank you for listening. And if you have any question, don't hesitate to send me an email. Thank you.
Thank you very much, Lisa. Fascinating talk. So we have time for a few questions. One online from AC. Sorry if I missed it, but did the added C13 labeled fatty acid convert to the sex factors?
So it's an experiment that we are thinking doing. We still haven't decided if we are going to do it or not, because labeled compounds are really expensive. So we're still not sure. But yes, the idea would be to synthesize linolenic and linoleic acid with using carbon-13, because the mass would be different. And then in theory, we would be able to trace back, if we feed them on the culture, we'll be able to trace which compounds are being produced from those fatty acids. Yeah, and find them on the mass analysis.
Okay, thank you. So I had a question in terms of, I was kind of fascinated by the fungal biofilm that you identified in vitro. I know you've done some proof of principle work in Planta, which you very briefly mentioned, but do you see any evidence of a similar biofilm being created in Planta? Have you had a chance to look at that?
Not yet, because I have to admit, when we go from a lab setting to a plant setting, even if it's in glasshouse and it's not field trial yet, the parameters are so different. So we have done a couple of experiments. We had good results, but we're still optimizing them. So for now, no, I was not able to do any microscopy on the plants that could show any biofilm production or anything like this. Not yet.
Okay. Does your fungus make a typical mat peptide pheromone? Mating type peptide pheromone.
I'm sorry, I can't answer the question. I don't recall reading about one, but I might be wrong.
Okay. Got one last question, I'm afraid. Very interesting, Lisa, from Maria. I used to work on Sex Factor with Alison Ashby and David Ingram about 20 years ago. Given how expensive SF is to produce, it would be a Sex Factor. It would be more cost effective to find some cheap fatty acids to spray. So hope it works. So I guess it's a comment, actually, sorry, rather than a question. Any AC, any data on the sex factor, what the sex factor receptors are yet?
So yes, we are also looking into this. Unfortunately, my PhD is ending in a year and 2 months, so I hope I will still be able to do this work. But we have some RNA sequencing data of the culture without the control, without sex factors, and the one with sex factors applied to them. And so I want to look at the expression of the receptors and hopefully try to knock them out using CRISPR and try to find out what receptors are linked to the sex factors. That would be really interesting.
Okay. One very quick last one. Andy Bailey asks, does the biofilm arise from lysis of the canidia? Any evidence for that?
I have no idea. I don't think so, because I think they covered the canidia, but sorry, I don't know.
Yeah, okay. And I'm sorry, we're going to have to move on. There is one last question. I'm sure that Lisa would be happy to answer that online. We've got to Linda, but we do need to move on. I'm really sorry.
Thank you.
Thank you very much, Lisa. Just because of time, I'm on strict instructions to keep to time. So we've got to move on to Jonathan Heal, who is in his second year of his PhD also at the University of Nottingham. And he works in the filamentous fungal lab of Paul Dyer. This project combines molecular genetics and food science. How genetic variation is influencing the flavor of blue cheeses, particularly with emphasis on understanding how it impacts bitterness. Jonathan, before starting his PhD, completed an integrated master's in biology, MBio, at the University of Oxford, and obviously because of the project, he's got a great interest in the fungal biology and its relatedness to food production and also ecological interactions. And his title of his talk tonight is A Bitter Tug of War, the search for a better blue cheese. Over to you, Jonathan.
Thank you, Mike. Everyone see that okay?
If you could put it into presenter mode. Yeah, there you go.
There we go.
Good.
Okay, so good evening, everyone, and welcome to my talk. I'm Jonathan, and today I'll be telling you about a bitter tug of war, the search for better blue cheese. So as a quick overview of the talk structure, first I'll be introducing blue cheese to everyone, so we're all on the same page, then talk about challenges with current blue cheese production, a little bit of detail on what I've been doing, and then some more about my future plans for the rest of my PhD. So to introduce blue cheese, well, firstly, we have to talk about what regular cheese is. So the definition of cheese I'm using is that it's a fermented dairy product made from milk curds. So milk, when you heat it or acidify it or add rennet to it, splits into two different parts, the solid curds and the liquid whey. And specifically, cheese has to be made from these curds. So here are a few things that you might call cheese. On the left, we have Icelandic skyr, we have mozzarella, we have goat's cheese. And on the right, we have ricotta, brunost, and vegan cheese. And of those, the only real cheeses are the ones on the left. They're made from the curds. Ricotta and brunost are made from whey, and vegan cheese, you probably don't need me to tell you that that isn't actually cheese. And historically, it's been used as a longer-term storage of milk. It doesn't tend to go off very quickly compared to actual milk, as I'm sure you've noticed. That gets lumpy in a week, but you can probably keep cheese in your fridge for a lot longer. We don't actually know when or where it was invented, but it has been consumed by humans for a very long time. And statistically, about 90% of Europeans like cheese, or at least okay with it. So we probably have a lot of cheese lovers among us today, which is fantastic. You're keeping me in work. Also, interestingly, if you don't like cheese, you probably have a family member who also doesn't like cheese. It seems to have a genetic component, though what the cheese-liking gene actually is, I'm afraid I can't say. So on to actual blue cheese now. This refers to when a cheese is matured with the blue mold Penicillium roqueforti. And maturing a cheese is necessary for the recognizable cheese flavors. So when you make cheese just from curds, at first it basically tastes of nothing. It needs to have some sort of microbial interaction in order to produce the typical cheese flavors. So normally it would be bacteria, but in the case of moldy cheese, you'd use a fungus. And yeasts are also very commonly involved. A different study found that about 66% of British people like or can at least tolerate blue cheese, though it is absolutely hated by about a third of us. It's a very polarizing food, as I'm sure you're aware. And there's been evidence of its consumption since around 800 BC. Some work looking into some human stool samples from around that time in archaeology found evidence of quite a lot of the mold and spores, suggesting that people at that time were eating it. And it's got quite a complex flavor profile due to the enzymatic activity of the mold, and I'll get on to more of that later. So this is a quick graphic just to show that blue cheese is enjoyed all over Europe, and lots of countries have their own varieties. So for example, we in the UK have Stilton, which you may have heard of, but there's also stuff like French Roquefort, Italian Gorgonzola, and loads of others. And they're basically all European. It's mainly a European food. Some of these are also very culturally important. They have a protected designation of origin, a PDO. So, for example, Stilton can only be made and properly called Stilton if it's made in Leicestershire, Derbyshire, or Nottinghamshire, hence its relevance for the place that I'm working at currently. So onto the mold. Penicillium roqueforti, which is reasonably closely related to the mold that makes penicillin, is an ascomycete mold, which is a type of fungus that it is. And it grows on a wide variety of substrates in the natural environment. It's very easy to find in the wild. But it's also very tolerant to extreme conditions. Now, you wouldn't think of food as being an extreme condition because we eat it all the time. But for a microbe, a lot of foods aren't very good for growth, cheese and bread included. In fact, Penicillium roqueforti is one of the few molds that can grow on bread. So if you ever had those annoying blue molds growing on an old loaf, it may have been Penicillium roqueforti. It's so tolerant, it can even grow on sourdough, which not many things can. Sourdough is a very hostile environment for mold. So if you want to make your own home cultures of the blue mold to try to grow your own blue cheese, one method of doing that is to take an existing blue cheese and rub the blue bit on some bread. And it will eventually look like this. You can crumble it up, add it to cheese, and try to make your own at home if you really want to. And of course, it's known for being blue, but specifically it's the spores that are blue. So it produces lots of those. And interestingly, it was quite recently discovered how the pathway of this works. So in fact, you can interrupt the creation of the blue color early on with various mutations to get yellow, white, red, green molds. So in future, you may start seeing different colors of blue cheese. I don't know if it will still be called blue cheese at the time, though. So having told you what it is, we'll now talk about some challenges for blue cheese production at the moment. So if you were reading the news at the start of this year, you may have seen some headlines about blue cheese being in peril. Traditional Roquefort and Camembert are under threat. So this is basically because the blue cheese mold, like many domesticated things, is primarily reproducing asexually, clonally. And when that happens, you get a lot of genetic degradation because the mutations are accumulating, they're not being filtered out as easily, and you eventually get a worse and worse product. So over time, eventually these things could become too terrible to actually make cheese anymore, and that would not be good. But fortunately, quite recently, how to make the mold reproduce sexually was discovered. And so the future of blue cheese is probably secure. This was a fairly recent discovery, and it's allowed us to make lots of new strains by crossing existing strains. You can cross existing commercial strains, but also put in some wild strains as well. In fact, this has been done, and it's been used to create a variety of new flavor profiles on different cheeses. And some taste testing of this found that some of them were really, really good. However, others were rather unpalatable, and we don't know why. Particularly, there was a problem with bitterness, which is what I'm personally interested in. That's what I'm looking for. So 3 of the strains listed on these graphs will come up later. A22 and B20 are both not very bitter strains that are supposed to taste quite good. And D18 is one that tastes very bad because it's extremely bitter. And those are 3 that I'm working on, along with a typical commercial strain, which we internally call 7488. So where does the bitterness in cheese come from? Well, there are a few possible causes. It could be down to contamination by a bacterium or even an overgrowth of things that are supposed to be there. Pictured is called Geotrichum candidum. It's another type of mold, and it's normally in blue cheese. But if it's allowed to grow too much, it produces very unpalatable cheese that you don't want to eat. So keeping that under control is necessary. Another possible cause of bitterness are the spores. So it's producing lots of spores, that's what turns it blue, but they are also thought to taste a little bit bitter. There's not really anything we can do about that. Maybe we could reduce the bitter compound being produced, but that's not something that I'm working on personally. What I'm interested in are called bitter peptides. So you may be aware that the main protein in milk is called casein, and casein is a protein that, when it's broken down, tends to produce lots of smaller fragments, peptides, which are very unpalatable. They're bitter, they're hydrophobic, and they bind to the bitter receptors on our tongue. And also, casein, when it gets broken down by rennet, produces big hydrophobic lumps that can then be broken down by the mold to produce more bitter peptides. So this is a real problem throughout cheese production. And yeah, you can get these small bits out of the main protein, and it makes something that you don't want to eat. And a final thing about blue cheese is the health consequences from eating it. So firstly, blue cheese is very high in salt because this is necessary to control the microbiome among everything else. Additionally, the salt does control the bitterness, as well as preventing the bitter molecules from being produced in the first place. It interferes with the bitter receptors on your tongue and makes you less likely to notice it. It masks it. However, eating something that's very high in salt is not good for you, so it would be good if we could replace it with a mold that is less bitter, and therefore we don't need to use as much salt. Secondly, you may have heard of what's called the French paradox, which is the idea that eating a French diet, it means that you're less likely to have some heart disease or things like that, despite the large amounts of saturated fat in the diet. Now, the French paradox, despite its popularity in certain dietary circles, is probably not real. It's more likely to be a statistical mistake. But there could still be health benefits from eating things like blue cheese because it has chemicals called andrastins, which are anti-cancer, and another called roquefortine, which is known to reduce cholesterol. So there could be benefits to eating it if we can make it a bit healthier. So now on to what I've been doing. To begin with, I've been looking at exopeptidase expression. So I'm going back to the enzymes now. In case you don't know, an enzyme is something that either breaks stuff down or makes something. That is an oversimplification, but it's the basics. And an exopeptidase specifically will break down peptides by targeting them from one or both ends. So they can be used to break down bitter peptides. In fact, this is done commercially. If you ever buy soy sauce, it's probably had exopeptidases added to it to make it less bitter. And so they will go through breaking off each subcomponent of the peptide, the amino acids, and then those themselves aren't bitter. And they might remove 1, 2, even 3 at a time. So our initial hypothesis is that higher exopeptidase expression must correlate with less bitter cheese. You'd expect that because that means there's more stuff breaking down the bitter compounds. So I had a look into it. So I looked into the genome and found 14 exopeptidases that are likely to be exported into the cheese matrix. They've got the signal peptide, which should tell the fungus to put them outside there. And I started looking at the RNA levels of those to see how high they were, and RNA levels correlate with enzyme quantities. In case you don't know, RNA is a bit like DNA. DNA is the center of information in the cell, whereas RNA is like a rough copy of that, which can be produced, sent out of the nucleus in the cell into the cellular environment. It doesn't matter if the RNA is damaged, whereas if DNA is damaged, that's a big deal. So therefore, you can get a rough number of RNA copies, and that should tell you how much is being made of the protein. However, the results that we got were about the opposite of what we predicted. So I used qPCR for this to tell me how much RNA was in the samples specific to each gene. On the right is an amplification plot. That doesn't matter too much. It's just to show exactly what I was doing. On the left is a bit more of a digestible graph. So to explain what that's showing, at the bottom, it's showing the expression levels of a particular exopeptidase relative to those in another strain. So at the left, we've got 7488, so over here. And you can see that 7488, which is a medium bitterness strain, is expressing this gene a lot more than A22 and B20, which are much more bitter strains. And we see the same thing as D18, and they're expressing this gene about the same as each other. So yeah, this was the complete opposite of what we were predicting with our hypothesis, which is fine. These things happen in science, but it's not what we were expecting. So we have to make a more complicated hypothesis. So this is where the tug of war comes in. We do still think that exogenous exopeptidases have an important role, but so must the proteases that are breaking down the protein and making the bitter molecules in the first place. Therefore, our hypothesis is that it's a balance between the expression of each that determines the bitterness levels. And this has been suggested in non-mold ripened cheeses. It's been demonstrated to some extent with bacterial ripened cheeses, where they only export proteases but keep the peptidases inside them. And if they die before they burst, then those peptidases will get broken down inside the cell and not released, and you get a more bitter cheese. Whereas if it's the other way around, if they burst before dying, or as they die, then they'll release the extra peptidases out and we get a less bitter cheese. But it's never really been demonstrated in the mold ripened stuff before, so that's what I'm looking at. And I found 5 exogenous proteases in the genome, and this work is still very early on, but early experiments do tentatively show support for this hypothesis. There does seem to be a correlation between expression levels of the proteases and how bitter a cheese strain is, but there's lots more to be done. Please don't take away from this talk that that is in any way definitely the case. I need to do lots more to check that it's okay, but it's still reasonably optimistic. And just as a quick diagram of what we're talking about here, so the protease activity, when it's greater, you get a more bitter, less palatable cheese, but when the peptidase activity is greater, the cheese is much nicer to eat. The other thing that I've been focusing on at the moment is trying to make a good cheese simulation. So blue cheese, as you may be aware, takes months to make, which is far too slow for a PhD project that only lasts a few years. You can't make that much, and it's also quite expensive. So I want to use a substitute medium for that. Currently, we use what we call milk media, which is milk, full-fat milk that has had salt and acid added to it in order to try to get the pH and the salt levels to about the same as actual blue cheese. But before I started, it never really been tested how well this simulates cheese. It should be similar in theory, but I wanted to test it before I used it for all of my work. So when you grow the mold in it, it starts to turn blue. It becomes that weird gray color by the end, and I wanted to know, does that taste like blue cheese? But obviously I didn't want to taste this. I'm sure you can see why. I doubt any of you would want to taste this, even if it might taste like blue cheese, however much you like it. So I had to come up with an alternative method. But fortunately, if you don't want to taste something, you can always get a robot to do it for you. And this is where an electronic tongue, or e-tongue, comes in. And this device has been used to taste things that people wouldn't want to taste before. So on the right, we have a picture of Professor Poppy working for Sainsbury's, and she was helping the e-tongue to determine what exactly in dog food dogs prefer, what type of tastes they like. So obviously, you can't give dog food to a person to tell you how it tastes and compare that to the dog's preferences. And I guess Mel Gibson wasn't around to help out at the time. So they gave it to the e-tongue and were able to find out what tastes there were in the dog food and how this correlated to dog preferences. So back to cheese. Here's the e-tongue doing some work for me. It's very noisy. You can hear it across the building when it's running, so sorry if you're struggling to hear me a little. I'll try to speak loudly. But the robot arm is dipping the probes at the end of it into the samples. Each of these has a membrane on it, which is specific to the types of taste thing that the sensor is detecting. So there's a bitterness sensor, a saltiness sensor, that sort of thing. The molecules will bind to the membrane. This changes the conductivity of the membrane, which can then be reported to the computer as a number. So it will report a value for bitterness, maybe 2.5 or something like that. And then you can compare this between different samples. So this is the data that I've got so far. It's still early days. I'm still interpreting it at the moment. But what I've got plotted here is called a principal component analysis, or PCA. The maths behind it is reasonably complicated, but simplifying it, what it is it's plotting all 8 of these different traits on there, and then each dot is close to other dots that have similar levels of those values. So I was looking at saltiness, sourness, bitterness, which all should speak for themselves, umami, which is a sort of meatiness or a rich taste, richness, which is umami aftertaste, bitter aftertaste, and astringency, which is the dry feeling in your mouth that you get after drinking tea or wine, and astringent aftertaste. And the dots that are together are very close on these values. So what you might be able to see on this graph, at the bottom, we have the dark blue circle, and that's all of my actual cheese samples. And then we've also got in red and dark green, the samples of the mold grown on milk. And in light blue and yellow, we have samples grown on milk media. And you'll probably see that the dots are closer to the cheese with the milk media than the actual milk ones, which is a good sign, but they're very much not overlapping. There is more work to be done in order to make the milk media better, I assume, but whether it's a time, growth difference, or something else that we need to change remains to be seen. So I'm briefly going to go over my future plans for the project as well. So firstly, I want to do proteomics. So I want to identify what the bitter peptides actually are, what their amino acid sequences are, and also look at enzyme levels in the cheese matrix. So for that, I want to see how much of each enzyme is actually present and whether this does correlate with the RNA expression work that I've done previously. This may also find new relevant enzymes if there's a lot of an enzyme that doesn't have a signal peptide out there, or if enzymes with signal peptides are not present, I can rule them out. I'd also like to do some time series work to see how this changes over time, either in cheese and or in the medium simulation. And I want to do some gene editing to artificially manipulate the enzyme levels to see how this changes the taste. Now, this isn't to make GM cheese for market. This is just to prove that enzymes are relevant by removing them or upregulating them and seeing if that can make a more bitter strain less bitter or vice versa. And finally, I'd like to compare wild and food strains to see how the expression levels differ on milk media, but also a more natural grass silage media, which is their wild habitat, or one of them, and see if we can predict a palatable wild strain based on protease and peptidase expression levels. That would be the fantastic end goal. And this is quite similar to some work that has been done recently, which looked at the entire genome. I'm just narrowing it down on specifically protease and peptidase genes. So I'd like to say thank you to the people who have helped me on my PhD journey so far, Professors Paul Dyer, Matthias Brock, and Ian Fisk, my supervisory team, PhD students, postdocs, and technicians in the lab groups that I'm in. You're all very wonderful. The BBSRC DTP for funding and training me, Mike and Niels for allowing me to use their strains, and Long Clawson, the cheese company, for the advice and support that they've provided. And if, in case anyone's interested in reading further, please screenshot these references and further reading, or email me and ask for them. They're good things to read. Thank you. Any questions?
Thank you, Jonathan. Thank you very much. So, time for a few questions. AC has popped one into the chat. So, are casein bitter peptides from different milk sources, goat, etc., approximately equal?
Ooh. Okay, that's a very good question. I'll admit I'm only focusing on cow milk at the moment, but it may well be the case that goat cheese and sheep milk cheese are different. I'd be happy to look into that for you. And if you could email me that question on the email address below, then I will find out. I'd be very happy to share. Sorry that I can't answer that now.
I have a little question. In terms of, trying to understand the balance between the peptidases and the peptide breaking down enzymes and the exopeptidases, obviously you're using RNA-seq and RT-PCR to get the quantification on that, but obviously in reality these things are actually proteins and there's not always a direct correlation. Has anybody looked at the actual stability of the different exopeptidases and the potential impact that that might have in the overall result in terms of the bitterness and what you're observing rather than just the expression levels at an RNA?
So that was actually brought up as a question right at the beginning of my project when I was pitching it in the first place. As far as I'm aware, no one's really looked into this. A lot of the exopeptidases and proteases, while they're in the genome, some weren't even known to be whether they were expressed or not. They were still listed as hypothetical proteins, things that should be proteins. So we don't know anything about their stability. I'm hopeful that maybe time series work and the proteomics, we'd be able to look into that. But as far as I'm aware, there's not really any information about that available at the moment. Maybe on one or two of the proteases that there's been a bit more work done on.
Okay, thank you. And Jen Cook's got an interesting one. If I experiment with wild molds, how can I tell if the moldy cheese produced is safe to eat?
So yeah, the wild molds, even if you do guaranteed have Penicillium roqueforti. It does produce some mycotoxins that you might remember hearing about in Alex's talk in particular. So I wouldn't advise experimenting with wild molds unless you're able to test for mycotoxin levels, doing some of those yeast assays, for example. Just in case you don't want to be eating something that would make you quite sick. Normally it would produce fewer of those in cheese, but I'd still recommend caution. Yeah, I wouldn't eat anything grown with a wild mold that hasn't been tested like that first, if I were you.
Okay, and Linda's a kind of a follow-up question to a certain extent. I guess if you take a sample from Stilton and you wanted to keep it and culture it fresh and to try and generate your own cheese at home, is there a best way to keep a culture fresh at home?
So you can grow it in bread and then keep that in the fridge for a little while. Obviously, it's not going to survive forever, but what you can do is keep growing it on more bread. Eventually, you will see degradation of the strain. I'm afraid that does just happen. But unless you have a -80 freezer or something like that, you probably can't keep it for a really extended period. But certainly you should be able to keep it for maybe even a matter of months in the fridge. I think it would probably be fine.
Yeah, follow up. Yeah, it might survive on frozen bed, perhaps. I don't know. You'd have to try it. Yeah,
Yeah.
it wouldn't be a end of the world to have to buy some Stilton every now and then, I suppose. Andy Bailey probably has to be the last question, I'm afraid, due to time, but Andy Bailey's got one. Does the bitterness also depend on the type of rennet that has been used to generate the curds?
Oh. So I'm afraid I've not really looked into things like that. The rennet causing bitterness is just something that I read a bit about. I don't know if people have really looked at different types of rennet changing the activity of the cleaving in particular. I wouldn't be surprised if it was fairly similar between different types of rennet, but I'm afraid I don't know, and I don't think people have really looked into that much. Sorry.
Okay. I think we're going to have to close questions there. So if all speakers would like to join me back on with their camera, it just leaves me to say thank you very much. I hope you all agree online that we've had 3 fantastic, interesting talks this evening. And thank you very much all. I really enjoyed those. Thanks.